Hey guys, welcome back. This is Chapter 9, DNA Structure and Replication. And we are beginning with Section 1, Experiments, Identify, and Describe Genetic Material. All right, so let's get going. So first we'll begin by talking about what is DNA. Well, you guys took biology before, so I'm hoping you remember just a little bit, because we're not going to go into super detail with some of these things, but you should sort of remember them. But DNA is our genetic material. It contains all of the information, the entire blueprint that the cell needs in order to synthesize proteins and replicate itself. So in other words, every single cell in your body has DNA in it, and that DNA tells it exactly what kind of cell to be, how to do its job, what kind of proteins to make in order to do its job. DNA stands for deoxyribonucleic acid. So some early experiments. Let's start in 1869 when Friedrich Miescher first isolated DNA and he named it nuclein. Now, at this time, people didn't know what the genetic source was. What is the molecule that is passed on with our code to make us, how we are. So when he first isolated this nuclein, he didn't know that it was a genetic code, but he recognized that it was a new macromolecule that had not been found before, but is present in cells. Skip forward to 1902, we have Archibald Garrett, which, well, first of all, right here we have Miescher, and over here on the right, that's Garrett. And Garrett was the first person to really link heredity to enzymes, which are a type of protein, and specifically to defects in the enzymes. In 1909, he published a book entitled Inborn Errors of Metabolism. So he was one of the first that really fought for the argument that the proteins have to be the genetic material, because the proteins are expressing our traits. When something's not right with the protein, the trait isn't expressed properly, so therefore it must be the protein that is our genetic material. Now, skipping ahead again, we have 1928, where Frederick Griffith identified some sort of transforming factor that transmitted infectiousness to bacteria. So this right here is Griffith. And this is what he did. So he took a rough and smooth strain of what I believe was similar to pneumonia, it was a pneumococcal bacteria. And first he took the rough strain of it. And with the rough strain, when he injected it into the mice survived. So it was non-virulent. The smooth strain, though, when he injected the smooth strain into the mouse got very sick and it died. So it was a virulent strain. Then he took the smooth strain, the one that killed the mice previously, and he killed them with heat. Then he took the dead smooth strain cells, injected into the mice, and they lived. So when the bacteria, these particular cells, these smooth cells, when they're alive, they kill the mice, but when they're dead, the mice survive. So then he tried the rough strain, which, remember, the mice survived that, and the heat-killed smooth strain, which, remember, the mice survived that by itself. But when he put them together and gave them to the mouse at the same time, he noticed the mice once again got sick and died, just like they did when they were injected with the living smooth strain. So Griffith came up with the idea that there has to be some kind of transformation factor going in, transforming factor, that the rough strain, which was non-lethal, transformed and became lethal when it was surrounded by the dead lethal strain. It took something from the dead cells and incorporated it into itself, which then turned the rough strain into the virulent lethal form of the bacteria. Sorry. In 1944, Oswald Avery, Colin MacLeod, and Macklin McCarty showed that this transforming factor was DNA. So pretty much they repeated Griffith's experiments, but what they looked at was more than just what was being, or more than just did the mouse live or die. They worked with the smooth strain, the virulent strain, but what they did was they actually took different macromolecules from it, destroyed those macromolecules, just that particular one, and then injected it into the mouse, and then looked to see what happened. So in one case, the polysaccharides, which are a type of carbohydrates, they destroyed the carbohydrate, the polysaccharides of the S strain. Now, these are all heat killed at this point. So these are the heat killed S strain. They destroyed the polysaccharides that are also found within this bacteria, injected into the mouse, still died. So the S strain, along with the heat killed S strain, along with the rough strain, which on its own is non-virulent. But remember, Griffith said when you mix them together, it turned the rough strain into the smooth strain. So they found that continued even though the polysaccharides were destroyed. All right, so the polysaccharide isn't what's being transformed in the rough strain. All right, then they took lapids and they destroyed those. Mouse still died. Okay, so it wasn't those or that particular molecule that was being changed in the rough strain. Then they destroyed the RNA of the S strain cells. Well, the rough strain still transformed, so, and the mouse died, so it couldn't have been that the rough strain were incorporating themselves. Tried it with protein, mouse still died. All right, so the rough strain wasn't taking the protein. Then they destroyed the DNA of the smooth strain cells. And what do you know? This time the mouse survived, and when they took blood samples from the mouse, there was no live smooth strain reported, which means that the rough strain wasn't able to incorporate something from the smooth strain in order to transform the rough strain into the smooth strain. Well, if it couldn't transform, it's because the molecule that it used from the smooth strain wasn't present. Well, the only thing that was destroyed at that point was the DNA. So that means that they showed it was the DNA of the smooth strain that the rough strain cells were incorporating it into themselves and allowing them to transform from the non-lethal rough strain into the lethal smooth strain. All right, DNA versus protein. In 1950, we have Alfred Hershey and Martha Chase, pictured down here on the left, and they used their famous blender experiment. And the reason why they did this was to confirm that DNA is our genetic material, not protein. Remember, way back in 1902, Garrett was the one who started saying that protein had to be our genetic material because protein expresses traits. When the protein is defective, the trait doesn't work. Therefore, protein has to be the genetic material. And there's been a huge back and forth ever since about whether it was this DNA or if it was protein. But in 1950, Hershey and Chase managed to put that to bed, that argument to bed, and they showed that it was DNA, not the protein, that had to be the genetic material. Now, what they did was they took bacteriophages. Now, what bacteriophages are is they are particular viruses that infect bacteria cells. Now, if you know how a bacteria or how viruses work, they attach to a host cell. In this case, the bacteriophage attaches to the bacteria cell. It injects its genetic material, which then hijacks the host cell and uses the host cell to make a whole bunch more of it. And then once there's a whole bunch of new virus cells made inside the host cell, that bursts open, and there's all these new viruses, or virus cells. So what they did was, they took the bacteriophages and they separated them into two groups. In one of them, they put these tags, these radioactive tags, on the protein of the bacteriophage. On the other group, they put tags on the DNA of the bacteriophage. We knew at this point that the virus injects its genetic material into the host cell in order to make more of itself. So what they were planning on doing is taking the bacteriophages, mixing them up with a whole bunch of bacteria cells, and then tracking where the radioactive tags were. Where the radioactive tags moved inside the cell, the bacteria cells, or did they stay outside? Well, with the bacteriophages, where the protein was tagged, when they were mixed with the bacteria cells and given time to infect the bacteria cells, what they noticed is when tracking the tags, none of the tags had gone inside the bacteria cells. They all stayed outside of the bacteria cells, which meant none of the proteins were injected into the host cells. When they tried this again, but with the bacteriophages where the DNA was tagged, they mixed them up with the bacteria, allowed them to have some time to infect the bacteria cells, and then they tracked the radioactive tags. And what they found is that a lot of the radioactive tags were now inside the bacteria cells. So that said that these viruses injected the tags into the bacteria cells. And since viruses inject their genetic material and the tags were on the DNA, it meant that the DNA had to be the genetic material that was being used and transmitted through with the virus. Around the same time, we have a chemist named Phoebus Labine, and he was the one who showed the different components. He discovered the different components of DNA. He found that it consisted of deoxyribose, some kind of nitrogenous base, and a phosphate group. And all of those in equal proportion. So it was a 1-to-1 ratio. For every 1 deoxyribose sugar, there was 1 nitrogen base and 1 phosphate. So if there were 10,000 deoxyribose, then there would be 10,000 bases and 10,000 phosphates. So this is just kind of showing what he discovered and what it means. It means that the DNA is made up of these subunits, which we now today, we call them nucleotides. where we have a sugar, a phosphate, and a base. Now, what was found is that there's actually 4 different bases. We have adenine, thymine, guanine, and cytosine. But for every 1 sugar, it comes with 1 phosphate, 1 base. Sugar, phosphate, base, sugar, phosphate, base, sugar, phosphate, base. And this is just showing you more the chemical structure of it, as opposed to just little easy to see diagrams. So thymine, cytosine, guanine, and adenine. All right. DNA structure. So in like the 40s and 50s, we started learning more about the actual structure of DNA. At this point, we knew it was a genetic material. We didn't really know what the structure of it was. Yes, way, way back, it was isolated, but we still couldn't really see it and figure out structure at that time. Now, Erwin Chargaff, when he did experiments and he collected data that showed the number of purines were pretty much equal to the number of pyrimidines. So the bases come in 2 varieties. There's the purines, which are these larger double ring structures, and the pyrimidines, which are these smaller single ring structures. And what he found is that the number of purines equals the number of pyrimidines. Specifically, when he analyzed the amount of each particular base, he found certain similarities. In yeast, 31.3% was adenine, 32.9% was thymine. Notice how close those are. They're very, very close. And then there's 18.7% guanine and 17.1% cytosine. So it's adenine, thymine, guanine, cytosine. Notice that the adenine and thymine are always very, very close to the amount of each other. Same with the cytosine and guanine. They're also super duper close. So not only does the purines and pyrimidines, they're equal to each other, but adenine, which is a purine, equals the number of thymine, which is a pyrimidine, and guanine, which is a purine, equals the number of cytosine, which is a pyrimidine. So this comes into play in the next set of discoveries when we find out that DNA is made up of 2 strands. And the reason why we have the same number of bases is because they actually match. So they're complementary to each other. Adenine will always bond with thymine, and cytosine will always bond with guanine. And that takes us to Rosalind Franklin, who used X-ray diffraction to deduce that DNA was a double helix. So this was in the 1950s, I believe, and she was working at a university in England, and using this technique, she managed to make this famous photo right here. It's called Photo 51. Now, it's not a photograph like what we think, what you take with your phones. What this is, it doesn't use visible light as much as diffraction, X-ray diffraction. And this is actually a photo of DNA. Now, the fact that there are 2 lines going like this means that there's 2 strands. And the fact that they crisscross like an X showed that it had to be a helical shape. So it's through her discovery that she managed to figure out that DNA was, in fact, a double helix. She did not get credit for that for the longest of time. In fact, it was the next 2 gentlemen, Watson and Crick, along with another colleague, Maurice Wilkin. They received the Nobel Prize for the discovery of the structure of DNA. Now, for the longest of time, we didn't realize Franklin's role in it, how she was the one who actually discovered it. But within recent years, within the past decade, a lot more of the credit has been given back to her. Sadly, she could not receive the Nobel Prize because she actually died not long after this discovery. We didn't quite realize at that time that x-rays were bad, and she ended up getting sick with, I believe, ovarian cancer, and she died from that not long later. Now, it's not to say Watson and Crick themselves didn't make an important contribution. They did not discover that DNA was a double helix as they claimed, but James Watson and Francis Crick were able to come up with the first 3D model of DNA. And they did this by using Franklin's data. So they actually got, thanks to Maurice Wilkins, they got a hold of her photo 51 and were able to create the actual first 3D model of it. And this actually is still important because once we were able to figure out the 3D model, the 3D shape of what DNA had to look like, it became very easy to understand how it's put together and also how DNA has to be replicated. So they did help in their discovery of the model of it, but they were not the ones who actually figured out it was a double helix. All right, and that concludes section 1 notes for chapter 9. I hope this was informative. If you have questions, hit me up in our Google Classroom or through email or in class. But until next time, you guys, just take care of yourself. Stay awesome. Stay curious. Stay safe and healthy, all right? All right, you guys, take care. Bye-bye.